I would like to start by thanking Vadim for the invitation. It's really an honor to be invited to this amazing meeting. I'm excited for all the talks and excited to be here to tell you about our work. Just to begin with an overview, my lab is very broad. We work on a lot of different topics. The overarching theme and goal is to understand how cells are organized in space and in time, and how that organization impacts cell function. So as you know, cells are not just a bag of nucleic acids and proteins, but they're highly compartmentalized and spatially organized. In my lab, we would like to be able to visualize that spatial organization at near molecular spatial resolution and understand its impact on cell function. And we work on this in the context of a few different biological problems. Today, I'm going to focus on our work on nuclear organization, in particular the organization of chromatin and how that regulates gene activity. So I don't probably need to introduce chromatin to this audience. As you already heard, DNA is this very large biopolymer that needs to be compacted inside the tiny space of the nucleus, and that compaction happens in the form of chromatin. This is a very complex structure that spans several length scales. These small length scales, between about a 10 nanometer and 100 nanometers, I think are particularly important for regulating gene activity. As you can imagine, if DNA is within these compact structures, its code is inaccessible to regulatory proteins. And so in order to be transcribed, the chromatin must unfold, making the DNA more easily accessible. And so in my lab, we are very interested in being able to visualize chromatin at these length scales in intact cells, in intact nuclei, and understand how these length scales are remodeled, how the fiber is remodeled in order to activate and repress genes. And doing this is not straightforward, especially in intact nuclei and using light microscopy, which is the main tool that we use in my lab, because these length scales are very small. And they're particularly below the spatial resolution limit of a normal light microscope, which is limited in its spatial resolution by the wavelength of light to about 200 to 300 nanometers. So to overcome this problem in my lab, we use super resolution light microscopy, in particular single molecule based methods like STORM and DNA paint. Again, I'm sure you're already familiar with these methods, so I'll go very quickly, just explain with one slide how they work using microtubules as an example. This is another structure my lab is very interested in. And here, what you're looking at is just a normal fluorescence image of microtubules in a cell. They form a complex network, but because of the spatial resolution limit, you can't really visualize individual microtubules within this network because they're very small and very close to each other. So what we do in super resolution light microscopy is to label the structure of interest with photo-switchable fluorophores. So these are fluorophores that can switch on and off between bright and dark states. And the idea is to switch off most of them into a dark state so that they're not visible. And then what we do is we excite just a few fluorophores at a time, such that their images are spatially segregated, and we can visualize them. So each one of these bluish-reddish blobs in that movie is an individual fluorophore. And even though they appear as large blobs because of the diffraction limit, nonetheless, we still can identify each individual fluorophore. And once we do that, we can also find the location of the fluorophore. We know that the fluorophore is at the center of that blob, and we can pinpoint that very precisely by finding the center position of the image. And we can put a dot to represent the position of each fluorophore. And over time, by activating new fluorophores, localizing them, and collecting more dots, we can reconstruct a high-resolution image of the underlying structure, which is no longer limited by diffraction. And so what I'm going to do today is tell you about how we are applying these tools to visualize chromatin structure in intact nuclei, and how that structure is remodeled in, for example, diseases. And so this work started many years ago in close collaboration with Pia Kozma's lab at CRG in Barcelona. At the time, my lab was also located in Barcelona, and we started collaborating. And Maria Aurelia, a former graduate student in Pia's lab and later on my postdoc, really spearheaded some of this early work. So what I'm showing you here is again, wide field conventional fluorescence image of H2B. This is one of the core histone proteins that forms the nucleosome. That's beads on a string-like structure that you saw in the textbook model. And this is a human fibroblast cell nucleus. Next to it, what you see is a super resolved image of the same thing. And this image has been color-coded according to nucleosome density. So blue corresponds to low density, more open regions of chromatin, and red corresponds to high density, more compact, more closed regions of chromatin. And I hope you can visually appreciate that by using super resolution microscopy, we do improve the spatial resolution. So now it becomes clear that nucleosomes are not just uniformly distributed everywhere inside the nucleus, but they form specific patterns at different length scales. And these images recapitulate some of the aspects of chromatin that we have known, for example, from electron microscopy. So what you can see, for example, is this ring at the nuclear periphery that is composed of high-density compact nucleosomes. And this is expected because we know that heterochromatin, condensed silence chromatin, is predominantly located at the nuclear lamina. And we can capture this in our images as this ring of high nucleosome density. But if we zoom into the nuclear interior, we also see some other patterns in these images at much smaller length scales. So for example, these red hotspots that sort of cluster together in close proximity. These are nanoscale in size. In fact, they are below the spatial resolution limit of our super resolution microscopy, so they're nanometric in size. And it took us quite a long time to understand what they represented. We had to develop quantitative approaches that allow us to estimate the number of nucleosomes within these resolution-limited nanoscale domains. And I'm going to skip many years of work in the interest of time and just sort of give you this model that we have settled on. Our quantitative analysis told us that these were small groups of nucleosomes. We called them nucleosome clutches, like egg clutches. They're quite heterogeneous. So in any given nucleus, you can have nucleosome clutches of varying sizes, consisting of somewhere between a few nucleosomes to a few hundred nucleosomes. So in terms of genomic scale, that's about 2 to 10 kilobases. And these nanoscale nucleosome clutch domains, clutches cluster together, as I mentioned, in close proximity, forming larger domains, which I call clutch domains for lack of a better term. But these larger domains are more in the size range of things like topologically associating domains and subdots, so hundreds of kilobases to megabases. This organization that I'm showing you here is quite a bit different from the textbook model that I showed you in my first slide, where chromatin is supposed to fold in a very ordered and hierarchical fashion from a 10 nanometer to a very ordered and beautiful 30 nanometer fiber. Nowadays, using both super resolution microscopy in my lab and other labs, as well as other technologies like Chrome EMT and Chrome STEM in Vadim Bachman's lab, we have come to realize that chromatin is actually more of a disordered fiber. And it's composed of these small groups of nucleosomes. Again, we call these nucleosome clutches. Vadim calls them nanoscale packing domains, so they go with different names. But we've, using different approaches, settled on a very similar picture of chromatin organization in the nucleus in the form of these nanoscale domains that can be quite heterogeneous. And we think that this is important for regulating gene activity because it provides more of a flexible template for regulatory proteins to act upon than this very rigid 30 nanometer-like fiber organization. And we do have some evidence that these are transcriptionally relevant. So for example, using multicolor super-resolution microscopy, we showed that the larger the nucleosome clutches and the more compacted the nucleosomes are, the more they exclude things like RNA polymerase II. So they're likely more transcriptionally silent regions of chromatin. Now, despite this disorder and heterogeneity, chromatin organization at these nanoscale length scales is cell type specific, and it can also be modulated using both chemical and mechanical cues. And I'm going to show you a few examples of what I mean by that. And so here, again, I'm showing you super-resolution images of chromatin in human fibroblast cells. This time, we actually labeled the DNA itself rather than the histones. So this is labeling using a nucleotide analog, EDC, and click chemistry. And we can use this for super-resolution microscopy. And these images give us very similar information to the nucleosome images. So once again, you can see those nanoscale packing domains appearing as high-density regions. And what I'm showing you here is on the top is a wild-type fibroblast nucleus, and on the bottom is a fibroblast nucleus that has been treated with TSA. TSA is a potent inhibitor of the HDAC enzyme. So when you inhibit HDAC, what happens is there is massive accumulation of acetylation, and this leads to opening of the chromatin and higher transcriptional activity. And again, I hope you can visually appreciate that these nanoscale domains change quite a bit upon TSA treatment. They become smaller and more uniformly dispersed inside the nucleus. Overall, chromatin density decreases, as we would expect. And you also see some loss of that peripheral heterochromatin. So there is a spatial remodeling of chromatin that we can capture using this microscopy. And in particular, we showed that the size of those nucleosome clutches decreases and they become less compacted. And so the packing of nucleosomes at that length scale is at least partially dependent on things like histone acetylation and the charge balance. Now, I also said that this organization is cell type specific. So again, I'm going to show you a couple of examples of that. First, in the context of induced pluripotent stem cells, or iPS cells. What Maria Aurelia did was take human fibroblast cells and overexpress the Yamanaka factors to reprogram them into induced pluripotent stem cells. And she made multiple iPS clones. So each clone comes from an individual reprogrammed human fibroblast cell. And then she visualized the chromatin organization of those clones. And she measured the size of the nucleosome clutches. So what you're looking at here, each dot in the plot is an individual cell. And we're plotting the average clutch size in that particular nucleus. As I mentioned, there's a huge heterogeneity, so we are kind of ignoring that and just taking an average number. And you are looking at here multiple clones, again, coming from individual reprogrammed human fibroblasts. And I hope what you can appreciate is the, variability in clutch size in between different clones. So some clones have, on average, larger clutches compared to other clones. And even within an individual clone, there can be quite a lot of variability in clutch size. And so we wondered if this was related to how pluripotent these individual clones are. And there are multiple ways of looking at their pluripotency level, and we've done many different assays. So for example, we injected these clones into mice and looked at the size of the teratomas they form. We looked at pluripotency markers. I'm just going to show you one plot that I think kind of summarizes the data very nicely. And what we did was we took these clones, we looked at the gene expression levels, and we compared those gene expression levels to known IPS standards and give them a pluripotency score based on that gene expression comparison. And what you see here on the x-axis is the pluripotency score, and on the y-axis is clutch size. And I hope you can appreciate this inverse correlation between clutch size and the level of pluripotency. So the more pluripotent a clone is, the smaller are the nucleosome clutches, corresponding to a more open state of chromatin. And again, this goes in line with the stem cells having more hyperdynamic, more open chromatin state, which we're here capturing quantitatively as the size of these nucleosome clutches. I will give you one other example in the context of embryonic stem cells, mouse embryonic stem cells, and their differentiation state. This is a similar plot as to the one I showed you in the previous slide. Again, we're looking at average clutch size, in this case in mouse embryonic stem cells. And these cells either have mutations or they have been cultured under different conditions that make them more or less pluripotent. So for example, if you culture ES cells in Twi-Lif, they're known to maintain their naive state of pluripotency. If you culture them in serum, they're primed to differentiate. And you can also take ES cells and treat them with retinoic acid to force them to differentiate into neuronal progenitor cells. So these cells have been ordered in order of decreasing pluripotency or increasing differentiation state. And again, you see that inverse correlation between clutch size and the level of pluripotency. The more highly pluripotent cells have smaller clutches. So we were very intrigued by this. We wondered if chromatin nanoscale organization can be used as a marker of cell identity. And in order to look at that, we didn't want to just look at clutch size because this is just one parameter, one very simple representation of these very complex images that give a lot of rich information. And so we've been collaborating with the lab of Jerome Solon in order to develop machine learning-based approaches that take these images and incorporate information at multiple length scales use that information to then cluster cells into cell states based on their spatial organization of chromatin. And I'll show you some results. So this is a confusion matrix where we've tried to classify ES cells grown in Twi-Lif versus neuronal progenitor cells, and the algorithm works really well in those two cell types. We can, with 100% accuracy, just from the chromatin spatial organization, classify them into the correct category. And we have applied this to more intermediate cell types as well with good results. So I've so far told you that chromatin, despite being heterogeneous, despite being disordered, is cell type specific. It's a marker of cell state, and it can be modulated with chemical cues like epigenetic drugs like TSA. Now, in the second part of my talk, I'm going to make a bit of a switch and tell you about how mechanical cues and biophysical cues impact chromatin organization. And this is a new, very exciting collaboration with several colleagues at Penn. So about 7 years ago, I relocated my lab from Barcelona to the University of Pennsylvania. And there I met a very active mechanobiology community. We have this Center for Engineering Mechanobiology, which is an NSF-funded center led by Vivek Shenoy. And they have kind of embraced me, and I've been having a lot of fun collaborating with them. So the work that I will present to you is in close collaboration with Suchin Hio's lab, Rob Mock's lab, and Vivek Shenoy's lab. They really got me interested in understanding how chromatin may sense and respond to physical and mechanical cues from the outside environment. So this, of course, has a lot of physiological relevance. As you know, different tissues in our bodies have different types of mechanical properties. So, for example, a brain tissue is much softer compared to something like bone. And cells that are in these mechanical environments sense and respond to those mechanical cues. We also know that the nucleus is not isolated from its external environment. It's actually tightly connected. On the nuclear envelope, we have something called the link complex. And the link complex is connected to the microtubule and the actin cytoskeleton. And so as the external environment changes and the actomyosin contractility of the cell changes, those forces are actually transmitted and sensed by the nucleus. They can, for example, put pressure on and stretch nuclear pore complexes, leading to shuttling of epigenetic regulators in and out of the nucleus. We know there are mechanosensitive transcription factors like YAP and TAZ. They respond to, again, mechanical cues by changing their localization between the nucleus and the cytoplasm. And so all these mechanical cues are sort of transmitted either in the form of physical forces or in the form of chemical signals into the nucleus. And they can potentially rewire the epigenetic state of the nucleus. We also know from many years of work from several labs, including at Penn, that if you take stem cells and you put them on a soft or a stiff substrate and you give them differentiation cues, they actually tend to differentiate into different lineages that resemble more cell types that are in stiff or soft environments. So we ask how much of that is encoded at the level of chromatin organization. So to address this question, we collaborated with Jason Burdick's lab, who develops these hydrogels. In this case, we're using MIHA hydrogels that can be made stiff or soft. And glass is our sort of baseline, which is much stiffer than both substrates. We took mesenchymal stem cells in this case, and we grew them on these different substrates and used super resolution microscopy to visualize their chromatin organization. And here I'm showing you some representative examples of what that looks like under different substrates. And again, I hope you can appreciate visually that we do see a dramatic remodeling of chromatin depending on substrate stiffness, and in particular on soft substrates. We see something very dramatic happening. Chromatin becomes predominantly localized at the nuclear periphery, forming this very thick layer of chromatin at the nuclear lamina. And the compaction level of chromatin also increases, both at the nuclear lamina, but also in the nuclear interior. If you look at those nucleosome clutches, they become larger and more tightly compacted. And I'm not just showing you a couple of cherry-picked examples. This is very robust, and we can quantify it. What I'm showing you here is the total amount of chromatin in the entire nucleus. So just looking at the signal coming from the entire nucleus. And that signal doesn't change, right? So we are visualizing the same amount of chromatin under the different conditions. But if we look at the nucleus at the the chromatin at the nuclear interior, the amount of chromatin at the nuclear interior goes down on soft substrates. And in accordance, the amount of chromatin at the nuclear border goes up. And we can also quantify, again, the compaction level of chromatin. Typically, we split chromatin into two compartments, the red regions, which is the more heterochromatic compartment, and the blue regions, which is the more euchromatic compartment. And again, if we compare stiff versus soft, for both the euchromatic and the heterochromatic compartment, the compaction overall goes up. So there is increase in chromatin compaction, chromatin condensation. And this happens dynamically over time. Again, Jason's lab develops these really cool hydrogels that start out soft, so 3 kilopascals, but we can stiffen them by cross-linking them with light and get them to be much stiffer. So what we can do is we can plate cells on the soft substrate and then stiffen them. Whoops. Very exciting things going on. Stiffen them and then fix cells at different time points to visualize their chromatin organization. And this is what I'm showing you here. So on the soft substrate, prior to stiffening, chromatin is more at the nuclear lamina, more highly compacted. Over time, it becomes more internalized to the nuclear interior and less compacted. And once again, we can quantify this. And these changes happen within a few hours. And interestingly, the compaction goes down within already a couple of hours upon stiffening, followed by a spatial remodeling from the periphery to the nuclear interior. And in accordance with that, epigenetic marks also change. So active marks go down on soft substrate, as we would expect, and silencing marks, in particular H3K27Me3, go up, particularly at the nuclear border on soft substrates. And if you look at EZH2, so this is the enzyme that deposits H3K27Me3, the amount of EZH2 also goes up on soft substrates in accordance with increased amount of H3K27Me3. So we wondered, is this methylation dependent? So to look at this, we use GSK, a potent inhibitor of EZH2. If we treat the cells with GSK and put them on soft substrates compared to untreated cells, we no longer see that spatial remodeling. So we can partially abrogate the spatial remodeling by inhibiting EZH2. So this is partially methylation dependent. And Vivek Shenoy's lab helped us use modeling to understand these results better. And in his model, he models chromatin as two compartments, euchromatin, heterochromatin. And the model incorporates interactions between chromatin and interactions between chromatin and nuclear lamina. And so in this model, euchromatin likes to interact more with euchromatin, heterochromatin more with heterochromatin and the nuclear lamina. So this leads to polymer-polymer phase separation between euchromatin and heterochromatin. But in addition to this chromatin interaction, he also includes rates of methylation and acetylation with the assumption that repressive methylation modifications will convert euchromatin into heterochromatin, and the active acetylation reactions will convert heterochromatin into euchromatin. So there's interconversion between euchromatin and heterochromatin. And if he assumes on soft substrates that there is higher repressive methylation rate, like we showed in our experiments, he can fully recapitulate our experimental data in his model. So these heterochromatic domains increase in size, and there's also more chromatin at the nuclear lamina, and that can be abrogated by abrogating the methylation reactions. And so this is partially dependent on the levels of methylation. Now, this was all cool. I'm a physicist by training. I like this kind of biophysical work. But I wondered, what is the physiological relevance? And my collaborators are in the orthopedic surgery department. So they convinced me there could be a pathological relevance. And they had access to human tenocytes isolated from healthy individuals suffering from tendonitis. This is inflammation of the tendon due to injury or overuse. And it actually leads to softening of the tendon tissue. So the tenocytes, which are the tendon resident cells, experience a soft mechanical environment in tendonitis. And then we also isolated aged tenocytes. And these are the super resolution images of chromatin. To my big surprise, tendonitis tenocytes had chromatin that resembled mesenchymal stem cells grown on soft substrates, with chromatin predominantly at the nuclear periphery and highly compacted. This was also very different from aged tenocytes. As you already heard, similar to senescent cells, heterochromatin is disrupted in aged cells, and particularly we see a loss of that peripheral heterochromatin. If we take healthy tenocytes and grow them on soft substrates, we can make them aberrantly remodel their chromatin to look more like the diseased tenocytes. And this was my favorite experiment. So if we take that stiffening hydrogel again and repeat it with healthy tenocytes, when the tenocytes are on the soft substrate, their chromatin is very aberrantly remodeled. But upon stiffening, they're able to respond to changes in their environment and remodel their chromatin back to a healthy-looking state. If you do this with the diseased tenocytes, they're no longer able to respond to changes in the mechanical environment. So they no longer remodel their chromatin, they have somehow lost that mechanoepigenetic memory. And again, these are all quantifiable. So I think I'm pretty out of time. I'm going to summarize. So I hope I could convince you that super-resolution microscopy is a powerful way of visualizing chromatin spatial organization. I showed you that chromatin folding is highly heterogeneous. This is also true for nucleosome dynamics. I didn't show you that data. But despite that heterogeneity, both the nucleosome organization and dynamics are cell type specific and can be modulated with both chemical and mechanical cues. And I showed you an example of how this remodeling is important in disease states where the mechanical environment of the tissue changes. For example, in tendonitis, where chromatin becomes more sequestered at the nuclear lamina, more highly compacted. We are, of course, wondering what are the implications of this in terms of gene expression, in terms of phenotype for the tenocyte cells. And these are some of the things that we're currently working on following up with. And with that, I'm going to finish by thanking everyone who've done the work. I think acknowledge everyone in my slides already. I'm really grateful to both current and former lab members, amazing collaborators over many years, both inside and outside of Penn. And just one final announcement. So I'm here with Marcus Woodworth, a very talented postdoctoral fellow in my lab. And he's presenting a poster on how he is using expansion microscopy, actually, to look at tenocyte chromatin in the context of native tendon tissue in tendon diseases. So I highly encourage you to go out and check out his poster. And thank you so much for your attention. I look forward to the discussion.